Okay, folks, let's get started. Happy Friday. Happy Mom's Weekend. How many moms? One? Two? Okay, I see two. Three? There's Mom. Mom, you have a note? You're late. What should we make moms do? Sing. You think moms should sing real loud? You know, as a matter of fact, I happen to have a song that's kind of special for moms. That'd be awfully embarrassing, though. It's actually something that moms probably wouldn't sing, but I think it might be appropriate for the students to sing to Mom. Okay? So, moms, this is for you. All right? So, and we're going to sing loud today, okay? You guys know the song Danny Boy? Yeah. Oh, Danny Boy. Da, 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 da. Okay, here we go. It's called Oh, Mommy Dear. Oh, Mommy dear, this is my biochemistry. I can't say. The problem's long, the course is really tough. That last exam, it really put the fear in me. I studied lots, I hope it was enough. But let's forget about it while you're visiting. And have some fun when we get out of class. Then when you're gone, I'll go back to my studying. That Kevin Ahern really, truly is an awesome guy. All right. Welcome, Mom. Okay. So, we've got a bit to do, so I've got a couple of announcements to make, and if you're really good, we might have another song as well, so we'll see. Wrong one. If I could learn to read my own outline. All right, so two things. One, we have an exam on Tuesday. People have asked when will there be a review session. I will have after class on Monday. I'm not sure if we can do it in this room. We got chased out last time, so I'll try to get that settled before we get chased out as last time. But we will have a review session after the class on Monday, okay? Material for the exam will cover through today. So wherever I finish today, that's where we will stop for the exam. Okay? Okay. Quiet group today. Everybody says, mom's here based on their best behavior. You know, moms, these guys are really bad, you know, when you're not here. I can tell you that, so. All right. So, last time I finished up on talking about attenuation. I know that's a very complicated process. Don't let it get bigger than the reality of it, okay? It's a mechanism for controlling whether or not an operon is synthesized. Keep in mind the big picture with that. If you're confused about it, come see me. I'll be happy to talk to you. But don't spend an inordinate amount of time on it, okay? What I want to do now is turn our attention to thinking about eukaryotes. And everything I've said so far about transcription has been about bacteria. So understanding a little bit about eukaryotes is important. And I'm going to talk about eukaryotes in fairly general ways. Eukaryotes, of course, are higher cells. And higher cells have a very big difference from the bacterial cells, as I've talked about before, in that they have their DNAs wrapped with these proteins called histones. And these proteins called histones block access of the RNA polymerase to the DNA so that it can make RNA. So there's a lot more orchestration. There's a lot more proteins that are involved in determining how transcription occurs, where transcription occurs, etc. There's also another level of complexity in that the eukaryotic DNAs are about 1,000 times longer than the prokaryotic DNAs. So finding the right genes is important and difficult. Okay? And last, when we think about higher cells, we're almost always talking about a multicellular organism. And so with a multicellular organism, we've got to think of differentiation. Okay? If we think about bacteria, all E. coli cells are identical, essentially. There's no one E. coli cell that does something, okay, I'm going to break down sugar, and the other one says, I'm going to break down fat. That's not the way it goes. But in the eukaryotic organism, we have, okay, this cell says I'm going to become a bone cell, and this cell says I'm going to become a blood cell, and this cell says I'm going to become a skin cell. Okay? Yet they all have exactly the same DNAs. Okay? So every cell in your body has the same DNA. So there has to be differences not in the DNA, but in the way that the DNA is expressed. So there's a lot more complexity to the control of those genes, because it's the control of those genes that determines, am I a blood cell? Am I a bone cell? Am I a skin cell? What kind of a cell am I? Okay? So those are very important differences. Now, we're just going to scratch the surface. We're not going to do anything deep. There are, entire year-long courses that cover eukaryotic gene expression, and we're not going to do that. But we're going to stand back and we're going to look at it from the top down. All right? So one of the big differences between prokaryotic and eukaryotic that we first notice is that in prokaryotic cells, there's one RNA polymerase. Only one. It doesn't even have a number. We just call it RNA polymerase. All right? We noticed in prokaryotic cells they had 3 DNA polymerases, but they've only got one RNA polymerase. In eukaryotic cells have 3 RNA polymerases. Okay? And they're roughly broken down according to function. Roughly broken down according to function. Okay? RNA polymerase I in eukaryotic cells mostly makes the ribosomal RNAs. Okay? Eukaryotic RNA polymerase II mostly makes the messenger RNAs. And in eukaryotes, RNA polymerase III mostly makes the small transfer RNAs. And there are some exceptions, but mostly pretty much covers what we'll be concerned about. Okay? RNA polymerase I makes the ribosomal RNAs. Okay? Okay. Now, there's a lot more complexity to the promoters in a eukaryotic cell. A lot more complexity. We saw there were basically 2, maybe 3 control sequences in the prokaryotic system. We saw the Pribno box at -10. We saw the -35 sequence. We also saw like the cap binding for some of these and so forth. Okay, there wasn't much that was there. In a eukaryotic, for a given eukaryotic gene, we might see 10 or 15 sequence elements that might play roles in helping to control when and how much of a given eukaryotic messenger RNA is made. Well, every time we have a sequence that's involved in control, we have to have a protein that's involved in binding to it. Okay, that's how that control is manifested. So we realize now that when we think about a lot of different sequence elements that's involved in eukaryotic gene expression, we start realizing that there's a heck of a lot of proteins that's involved. And in fact, it's quite different than prokaryotic. In prokaryotic transcription, RNA polymerase had that sigma factor, right? And the sigma factor determined what it bound to, and that was basically it as far as we were concerned. In eukaryotic transcription, that is the RNA polymerase involved in eukaryotes, usually the RNA polymerase has nothing to do with the binding. The binding is started by a series of other proteins that all build what essentially is a complex that the RNA polymerase comes in and then gets started with transcribing. So the setup is considerably different. Okay? We can think of the RNA polymerase in eukaryotes as sort of like a queen bee, right? The queen bee doesn't have to do anything. The queen bee comes in, the nest has been made, everything is set, the queen bee sits there and is the queen bee. That's what's happening in eukaryotic transcription because all these other proteins are coming in, getting everything set up, and that queen bee, being the RNA polymerase, comes in and says, oh, you want me to make RNA here? Oh, okay, I'll go and do it. And that's how it functions. The nature of the control sequences in eukaryotes are considerably different as well. We think of those promoters, the promoters that you saw in prokaryotes, at most they were 150 base pairs away from the transcriptional start site. Okay? In eukaryotic cells, they may be several thousand base pairs away. Okay? Several thousand base pairs away. Well, that's possible partly because remember that the eukaryotic DNA is much bigger, so things are much roomier. That's possible there. It's also possible because we have these many proteins that play these functions. All right? one of the proteins that we see in eukaryotic cells that we don't really see much of the same sort of thing in prokaryotic cells is something called enhancers. Okay? So enhancer sequence, so I use two terms here. I'm a little different than some people. I call enhancers the proteins that bind to enhancer sequence elements. Okay? So we have, you know what a sequence element is? That's something that a protein binds to. And so I call enhancer the protein that binds to an enhancer sequence element. All right? What is an enhancer sequence element, and how is it different from the control sequences that we've seen so far? Okay? Well, one of the ways in which that is different is, as I've said, these can be thousands of base pairs away. Okay? They get bound by an enhancer, and what the enhancer can do is actually help by bending the DNA to get the RNA polymerase positioned right for starting transcription. So here we have a good example of that. Here's an enhancer, and they use enhancer. It's actually enhancer sequence element. Here is the enhancer itself. So like I say, my designation is a little bit different than theirs. Okay? The enhancer binds to the enhancer sequence element, and this guy's a long ways away from the transcriptional start site. But what this can do is facilitate the bending of DNA to bring the enhancer and the machinery necessary to start transcription together. And when I say machinery, I'm talking about these proteins that are helping make that nest for the queen bee. Okay? So this helps us to understand how those control sequences can actually be thousands of base pairs away, but still affect the transcription of a gene. Okay? Okay, so enhancers really are fundamentally different types of control elements in eukaryotes and prokaryotes. Well, why are they important? It's a very good reason that they're important. Okay? Let's think about the bone cell versus the blood cell. Okay? The bone cell, or maybe even think better about a muscle cell. Okay? A muscle cell compared to a bone cell. All right? What does a muscle cell do? Well, a muscle cell does work. A muscle cell contracts. Because of that contraction that muscle cells do, we have movement. Okay? So understanding what a muscle cell needs helps us to see that, wow, muscle cells have certain proteins that have to help the muscle cell to contract. A bone cell doesn't need that. Okay? So if the cell's going to be efficient, I want my muscle cell to make this contractile protein. I don't want my bone cell making it because it would be useless. Plus, I wouldn't want my bones contracting. That'd be kind of a scary thing, right? Okay? You sort of shrink your skeleton, right? You'd be like those trucks that go down, you know, and so forth, but they're not supposed to. That'd be kind of, okay. Anyway, you wouldn't want to have your bone cell doing this, all right? Well, it turns out that enhancers are tissue-specific. That is, enhancers are made in certain tissues, and they bind to specific sequences of specific genes, and in other tissues, they're not made. So I might have some enhancers that are specific for muscle cells, and they activate a class of genes for muscle cells, but bone cells wouldn't have those same enhancers. So we can start to see very easily now how it is that differentiation can lead to control of different genes. So these really match themselves very, very nicely. Okay? Very, very nicely. So that means, therefore, that the scheme for getting the right genes made in the right tissue, it's complex, but it's understandable. Okay? A bone cell is going to have some things that it needs that a muscle cell won't have. Okay? Bone cell is going to be making bone, right? You don't want your muscle cells making bone. So bone cells are going to have their own enhancers for a different class of genes that they're going to be expressing. So enhancers, the bottom line to take away on enhancers is that they give tissue specificity. That's a really important thing. They give tissue specificity. So now we can start to see how the bone cells or muscle cells or blood cells make the unique set of proteins that they each need for their own particular function. Okay. We can skip that. Let's skip that. Okay. So we've been talking a fair amount, or I've been talking a fair amount. We don't talk. I talk. About DNAs that bind to proteins. Okay? And DNAs binding to proteins are really critical for the types of things that I'm talking about here, because all of the control of the transcription happens because of different things that the different proteins are doing, whether they bind, don't bind. You've seen proteins that can activate transcription. You've seen proteins that can inhibit transcription. Okay? And how do they do that? Well, it turns out that DNA binding proteins have some specific structural features that are common to them. Okay? They're called binding domains. So when we talk about a domain in a protein, that's a fairly specific term. Okay? It means a general thing, but the specific term says that a domain is a structural component of a protein that's common across many proteins. All right? So I might have a domain of a protein that really likes to grab a hold of those phosphates in DNA. It makes sense for one DNA binding protein I have to have that. It might make sense for other DNA binding proteins to have that, so that I don't have to reinvent the wheel each time I want to have another protein to bind to DNA. So that proteins have evolved common features, and they share those features. Somebody asked me last time about, well, how is it that introns are important? Why are eukaryotic genes in pieces? And it turns out that over evolutionary history, the pieces really make sense because they allow the cell to swap components. Let's imagine that I've got a component that's on this piece right here that binds to DNA. If I duplicate that piece, now I have two different proteins. This one over here can evolve into another protein that can bind to DNA. I have two different proteins that bind to DNA, but they use the common structural feature. Okay? That really turns out to be useful over evolutionary history. All right. Well, so binding domains, there are several that we talk about in this class. I think I maybe even mentioned a couple of them earlier briefly. I'll mention them again. One is called Helixturnhelix. And you can see that this Helixturnhelix is a protein structure. There's the helix, there's the turn, there's the helix. Okay? And if you look at that structure, you see how very nicely it fits in what we call the major groove of DNA. Major groove of DNA is that big gap between the strands. So the major groove is this portion. The minor groove is this little narrow portion right here. Okay? So Helixturnhelix fits nicely in that major groove. And what these proteins do, it's interesting. You might wonder, how does a protein know where to bind? It turns out that the proteins are actually reading, I'm literally saying that, literally reading the hydrogen bonds that are in there. So they can position themselves and go, oh, this is a GC base pair. This is an AT base pair. This is a TA base pair. Okay? And they can read those and, as a consequence, know where the proper place for them to bind is. So that's kind of a cool feature. So Helixturnhelix is one type of DNA binding domain. Another is called zinc fingers. Zinc fingers I've talked about before. They have a structure kind of like this, where we have a couple of cysteines, a couple of histidines, and they coordinate a zinc ion in the middle. And in the process of doing that, they loop out this finger. Well, this finger can be used to stick into the DNA and, again, read those base pairs at the place where the finger is sticking in. It's called a zinc finger. And you can see different structures for it, but they're all common in having this finger of some sort that's sticking out. Zinc finger. Okay. And let's see. Is that what you referred to as metal interactions? No. No, that's not what I referred to as metal interactions. No. Yes?
What is the reason for the zinc fingers?
The reason for the zinc fingers is it's just a common structure of proteins that bind to DNA. Okay? So it's just a common structure of proteins that bind to DNA. So commonly they will have things like a zinc finger. And you can see these fingers sticking out. And again, we see them mostly here in the major groove where they're interacting with the DNA. Last, we have what are called leucine zippers. And leucine zippers, actually, I won't talk about leucine zippers. They get a little confusing, but they're kind of cool. If you want to learn more about leucine zippers, give me a holler and I'll tell you something about them. Okay. Well, the last thing I want to say actually has to do with how we modify RNAs. And the modification of those RNAs happens for each of the different types of RNAs that I've described so far. Transfer RNAs are fairly commonly modified You don't need to memorize what the exact modifications are, but I think understanding in general the modifications that happen are important. When we look at the DNA from which the transcription to make the transfer RNAs and the ribosomal RNAs occurs, that is, these are encoded in the DNA, and the RNA polymerase comes along and transcribes them. If we look at those regions, we discover that the genes for the tRNAs and the ribosomal RNAs are long clusters. They're all clustered together in a long fragment. Okay? Well, if we have all of the tRNAs and the ribosomal RNAs made onto one long RNA, that means that if we're going to have individual ones of those, we've got to chop them up appropriately, process them, and so forth. So there's a lot of processing that goes on to convert that long segment that has all the ribosomal RNAs and tRNAs into it into smaller pieces. And that's part of what you're seeing right here. You can see here are a couple of transfer RNAs that have been synthesized. You can see the sort of general structure of the transfer RNA there. And these guys are still linked to each other. They have to be broken apart, as you can see. So you see an endonuclease that comes along and cuts it. And when it cuts it, it splits it into two pieces that you can see here. And those two pieces aren't quite there. They finally have to be processed a little bit further. This guy's a little bit too long. This guy's a little bit too short. It has to be modified. And we put some various nucleotides on the end and finally end up with a final transfer RNA that occurs. So processing of these, and this happens also for the ribosomal RNAs, but processing of the RNAs out of bigger pieces into smaller ones is a very important part of making the final mature ones. When we talk about eukaryotes, okay, and by the way, the processing that I just described to you is prokaryotes and eukaryotes. It's not one or the other. Okay, it happens in both. But there is something that's specific to eukaryotes that I do want you to know. All right? And that has to do with messenger RNAs. So messenger RNAs are handled quite a bit differently in eukaryotes compared to prokaryotes. If you recall, in prokaryotes we had no nucleus. We had transcription that could be followed very closely by translation. Okay? We also think about the life cycle of a prokaryotic cell. An E. coli cell can divide, if there's food, in every 20 minutes. Okay? And so one of the things that prokaryotes do is they have to control how much messenger RNA they have as well. Right? So if you can imagine if your lifespan was 20 minutes, all right, how many changes your cells would have to go through in that 20 minutes, right? I mean, some of the cells, for example, in our eyes, we are born with and we die with the same cells, you know? So we might have cells in our eyes that live for 80 years or 100 years, okay? So that's a very different kind of a situation than a bacterium has. A bacterium goes through a 20-minute life cycle, basically. So when we look at messenger RNAs in bacteria, what we see is that they are broken down much faster than they are in eukaryotic cells. The half-life of a messenger RNA in prokaryotic cells is on the order of a few minutes, meaning it's only around for a few minutes before it gets chewed up. And yes, cells do chew up their messenger RNAs. So for a few minutes, and that's a long time for a messenger RNA. In a eukaryotic cell, it varies quite a bit, but it wouldn't be uncommon for a messenger RNA to be stable for 24 hours. Okay? Well, how is it and why is it that they're more stable in a eukaryotic cell? Well, the why I've sort of told you already, that is that a eukaryotic cell is not dividing as rapidly as a prokaryotic cell. But how is a bigger question. All cells have enzymes called RNases, R-N-A-S-E. These are enzymes that break down RNA. They're part of that turnover process that cells have to go through. Well, why don't the RNases break down the messenger RNA? Well, they do, but not very efficiently. Okay? So when we look at eukaryotic cells, they've got two things that slow down the breakdown of eukaryotic messenger RNAs. This is why the eukaryotic messenger RNAs are around for a longer period of time. At the 5' end of eukaryotic messenger RNAs, there's an unusual structure called a cap, C-A-P. And the cap is basically a modified guanine that is attached to the 5' end. So here's the 5' end. This was the very first base that was put into the messenger RNA as it was being made. There's the second, the third, the fourth, et cetera, down here. Okay? There's the first base. Well, what's that? That's the cap. All right? The cap, as I said, is a modified guanine nucleotide. We don't need to worry about what the exact modification is. But you do need to know the unusual linkage. This is the only place in nature this linkage occurs. This is a 5' to 5' linkage. That's carbon number 5, and that's carbon number 5. Okay? That cap does two things. One, it protects from a nuclease coming in and chewing it up. So the nuclease can't start at the end and start chewing inwards because the cap stops it. The nuclease can't touch it. The second thing is that the cap helps the translation process, which we'll be talking about next. So the cap helps in the translation process. Okay, so that takes care of the 5' end of the gene. What about the 3' end of a messenger RNA? And at the 3' end in eukaryotes, we also see a modification. And it's kind of cool. Okay? Yeah?
So you said you led into it with two things slow down the breakdown.
Yes.
So the cap modifies the guanine and the 5' end.
Cap modifies at the 5' end, and the other one is at the 3' end, which I'm getting ready to say. Okay? The other one's at the 3' end. So the 3' end, that's the other, and that's your question. At the 3' end, the other protection against breakdown is something called the poly-A sequence. What is a poly-A sequence? At the 3' end of eukaryotic messenger RNAs, there's about 200 to 300 A's that are placed at the end. A. Okay? Well, what does that do? Well, it doesn't stop the nuclease. It doesn't stop it. But nucleases work by starting at one end and breaking it down one nucleotide at a time, chewing in, chewing in, chewing in, chewing in. Right? The longer the number of A's that the nuclease has to go through, the longer of a time it's going to take to chew through it, and the stabler that messenger RNA is going to be. Okay? We can actually measure how long a messenger RNA will be around by measuring how many A's are on the end of it. Okay? So kind of like the telomeres that we talked about with the chromosomes, where we had that fuse that was the bomb was going to go off, right? That's what the A's are on a messenger RNA in eukaryotes. It's a fuse. Longer gives more stability. Question?
What was the grouping of A's called?
I'm sorry?
What was the grouping of A's called?
The grouping of A's was called a poly-A sequence. Poly, P-O-L-Y. Yeah.
So does it just take a long time to get through because the AT bonds are stronger?
There's no. Remember, this is single-stranded, so it's just A's. There's no AT bonds at all. That would be if we had base pairs. So these are just RNAs with a long string of A's at the very end. Okay. Okay. So kind of cool things. Now the other thing that happens, and that's when this thing, I don't have a figure, but that polyadenylation that's right there, that's what poly-A means. Polyadenylation, poly-A. You can call it poly-A. All right, well, I've already talked about splicing briefly. I'll just briefly mention it one more time just to refresh your memories. But in eukaryotic cells, but not in prokaryotic cells, genes are in pieces. Okay? The relevant pieces for the final product are called exons. You can see them there in light blue, 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 blue. And in the process of splicing, what happens is the intervening sequences called the introns are removed. You can see them being removed right here. And the blue pieces are joined together. So in going from up here, the DNA to the RNA to the finished product RNA, we have gone from this guy to this guy, and you see the yellow sequences are gone. That splicing, it's called splicing. That process occurs in eukaryotic cells. It does not occur in prokaryotic cells. And interestingly, when we look at a protein and we look at, well, here's this exon right here. Where is that in that protein? We see that exons usually correspond to functional domains. So that DNA binding part might be one exon, linked to another DNA binding part over here on a different exon. Okay? So now we see that in splicing, the real advantage over evolutionary time is that cells have the ability to mix and match exons and create brand new proteins very simply. It's a very cool way to make proteins. What's that? Kind of like a Mr. Potato Head. I guess, yeah. So you can take and mix the ears. With Mr. Potato Head, I always made these bizarre faces, though I never did very good with that. So I don't know. Okay, questions about that? Okay. Let's see. And what's that? Okay, splicing occurs in a bunch of ways. All right. Let's start protein synthesis. And I have to leave early today, so that'll be good. And we'll get started with it, and then we'll finish with a song and a blast, okay? All right. So translation is the last part of the central dogma, all right? So we said DNA replication, transcription, and translation. So translation is the synthesis of protein using messenger RNA as a template and the genetic code. Yes, ma'am?
So everything today you're covering on the exam?
Everything today I'm covering on the exam. I'm sorry?
Including this little bit, even though you're...
Including this little bit. Oh, let's not include it. Okay, we'll stop it right there. How's that? One less thing that'll mean just that much more for the next exam, right? So, okay, so we'll stop the material for the exam with transcription. How's that? Okay? Yes, okay. All right. Now you can turn your brains off while I say this, right? All right. The process of translation is critical, obviously, because without translation we can't make proteins. Without proteins, we are dead. Many antibiotics are designed to kill specific organisms by stopping their ability to translate. So if you can find a difference between the way a bacterium makes protein and the way a human makes protein, then you can design a drug that will stop the bacterial function without harming the human function and therefore kill bacteria and not affect humans. That's the aim. Okay? Well, finding those differences, I will tell you, is not a trivial thing, because the overall general processes are very, very similar. Right? Nonetheless, people have found differences between them. There are really 4 things that happen in the process of translation. I told you before we had initiation, elongation, and termination, both in translation as well as in transcription. Okay? In translation, we also have this thing called amino acid activation. What the heck is that? All right? Well, if you recall when I showed you the structure of a transfer RNA, we saw that cloverleaf structure. Remember that? Okay? We had one end where I said the amino acid was attached to the transfer RNA, and the other end I called the anticodon. Anybody remember that? Okay? The activation is the process whereby that amino acid gets attached covalently to that transfer RNA. Okay? So amino acid activation is when the amino acid is covalently linked to the transfer RNA. Okay? All right. Well, how does a cell know how to do what? Well, all the rules that govern translation occur through what's called the genetic code. We've all heard of the genetic code, and I'm sure you got this in your basic biology. But the genetic code is the language of cells. It's the language. Okay? We know that the genetic code is written in 3-letter increments. All right? The 3-letter increments of the genetic code are called a codon. So the genetic code is written on the screen, and the genetic code is what we call universal, meaning that the bacteria that are in your gut are using exactly the same genetic code as you are. In fact, virtually every organism on the face of the earth uses the same genetic code. There's only very minor differences in a few places. 99.9999% all use the same genetic code. And the only places where there's differences, they're very minor. One change, for example. Okay. Well, the codon is a sequence that we see in the messenger RNA. Remember that. The codon is a sequence we see in the messenger RNA. The anticodon is the complementary sequence that we see in the transfer RNA. So we have codon in the messenger RNA. We have anticodon in the transfer RNA. Okay? If I wanted to look at a messenger RNA and say, what's the sequence of amino acids that corresponds to this, I would take the sequence of bases that I have in the messenger RNA, and I would use this information like a decoder ring to tell me what amino acids corresponded to. So, for example, if the first 3 bases that I wanted to translate were CUU, I would say CU, and then U would tell me it's leucine. Followed by an ACU would go ACU threonine, okay, etc. So that's how I would read the genetic code. So it's because of the genetic code, because of transfer RNAs, that information in transfer RNAs is converted into protein sequence information. Information in messenger RNAs is converted into protein sequence information. Very, very, very critical. If we look at the genetic code, we see something interesting. Okay? 3-letter code, but look at that 3rd letter. Valine can be written, it can be specified by GUU, it can be specified by GUC, it can be specified by GUA, and it can be specified by GUG. I think you can see a pattern. We could actually write the code for valine as GUX, where X corresponds to any nucleotide. Many, but not all, amino acids have that same, those codons have the same feature. Let's look at leucine. CUU, CUC, CUA, CUG. Okay? Where we see that there's a couple, we see something else interesting. Here's aspartic acid. GAU, GAC. Notice GA pyrimidine. For glutamic acid, GA purine, GA purine. Okay? Now, what does this mean? Well, why do we have this? This is what's called redundancy in the code. Redundancy. Okay? It means that a given amino acid can have more than one codon that corresponds to it. Some only have one. Look at methionine. The only one that makes methionine is AUG. Look at tryptophan. The only one that makes tryptophan is UGG. Okay. Well, methionine turns out to be a very important codon. It's one that I'll expect you to know the sequence of, because AUG is how almost all proteins start coding. Usually the very first amino acid put into proteins is methionine. So usually the very first codon that's translated is AUG. Okay? So we have something called a start codon. That's what AUG is. We also have 3 codons that are called stop codons. They tell the ribosome where to stop making protein. They correspond to UGA. So here's UGA. That's a stop. They correspond to UAG. I'm sorry, UA. UA. I can't read my own thing. UAG and UAA. Okay? The 3 stop codons, whenever the ribosome encounters those, it says, oh, I know I've finished doing what I need to do. Okay? Questions about that? Everybody's ready to finish for the week. Should we finish with the song? Okay. So I've got, this is a very fun and raucous song, and I usually lose my voice singing it, so you'll have to help me today, okay? Everybody like the Village People? Okay. This is a song about DNA. It's called BDNA. Are you ready? Phosphates are in nucleotides. I say phosphates cover bases inside. I say phosphates span the 5 and 3 primes. There's no need to be real mixed up bases. Carry info, you see. I say bases are all complementary. I say bases like A, T, G, and Z. They have got to be all paired up. Boom, 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 boom. It's fun to play with some bDNA. It's got a boatload of GCTA. It's got everything a polymerase needs when you melt all the A's and T's. It's fun to play with some bDNA. It's got a boatload of GCTA. You can make RNAs with a polymerase just by pairing up U's with A's. I'm terrible. Proteins full of amino A's. I say proteins come from mRNAs. I say proteins require tRNAs. There is more you need to translate codons. Like our friend UAC, I say codons come in clusters of 3. I say codons have one base wobble E. Now you can go forth and translate. Boom, 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 boom. It's fun to play with some bDNA. It's got a boatload of GCTA. With those hydrogen B's and right-hand helices, anti-parallel 5s and 3s. And I'm losing my voice, so I'm not going to go any further. All right. See you guys on Monday. Hi. Yes, ma'ams.
I have like multiple questions.